Hello, everybody. In this lecture, we're going to discuss viral genomes, which is the missing piece to viruses that we haven't discussed yet. So let's get right into it. By the end of this module, you should be able to differentiate between the types of viral genomes and explain the central dogma as it applies to viral genomes. So most viruses have a very small number of genes, less than 300. They're going to code for things like the capsomere proteins that make the capsid. They're going to code for any specialized structural factors, spike proteins, etc. And they're going to code for any enzymes necessary for replication of the virus. However, the genomes can be very different from the genomes of living organisms. So all living organisms have DNA genomes. They're all double-stranded. They follow the central dogma of DNA to RNA to protein. However, viral genomes can be formed as either RNA or DNA. They can be single or double-stranded. They can be single or segmented, and they can be circular or linear. And so it can be quite a hodgepodge of variation. And keep in mind that the goal of all viruses is to get the host to make proteins so you can make more virions. And so here's kind of an example of how we get, to mRNA, right? So there are these, you can have double-stranded DNA, which is going to follow just like normal DNA, right? And so you get our mRNA. Once you have mRNA, you can make your proteins. However, when you get into some of these other examples, there can be some different intermediates and things that affect the way that transcription occurs. All right. So as I was saying, with double-stranded DNA, this is just like the host DNA. So it's going to follow the same process of you just have a DNA-dependent RNA polymerase, and then that's going to make mRNA, which is going to go to your ribosome and get translated. Now we have the potential of having a negative sense single-stranded DNA. And so this, when you have these single-stranded DNAs, you're going to form a double-stranded DNA intermediate. The negative sense is the antisense or the template. So the negative sense DNA is the thing that gets transcribed, but it always goes to a double-stranded DNA intermediate. And the reason for that is, one, that is going to directly feed into DNA replication, which is going to produce both the positive and negative strands. And then those negative strands can be used both for packaging into new capsids and for making mRNAs. Now, the positive sense strand of DNA, same idea. It's going to go into the double-stranded form, so it can be replicated. When it's replicated, though, the negative sense strands can make the new mRNAs, while the positive sense strands can be packaged into new viral capsids. When we have single-stranded RNA viruses, it's a little bit different. You can have your positive sense RNA viruses. If you have a positive sense RNA, so this is where it gets a little bit confusing. The negative DNA makes a positive RNA. The positive RNA is the same thing as mRNA. And so it can be directly translated by the host ribosome. It also needs to be replicated, though, right? And so to do that, which we see over here, is we can get the positive sense RNAs can be directly translated, and then they can be replicated. The way that they're replicated, they need a special enzyme. And so to get that special enzyme, you have to translate the message. It produces this RNA-dependent RNA polymerase. Most of our RNA polymerases are DNA dependent, right? So that's what we see when we make a normal transcript is we use a DNA dependent RNA polymerase, which means that it makes RNA from DNA. In this case, we're going to make RNA from RNA. So we need an RNA dependent RNA polymerase. I know that's a lot of letters, but if you think about what's going on, it should make at least some sense. And so in this case, those positive sense strands can also undergo additional translation if necessary, or be packaged into the capsid that can be released. Now, on the other hand, we have our negative sense RNA genomes. And those negative sense RNA genomes are going to need to use a RNA-dependent RNA polymerase to make copies before anything else can happen. And so these oftentimes are going to package that RNA-dependent RNA polymerase in the capsid itself, right? Because when it comes out, this is not going to be able to be transcribed, right? And so you need this enzyme that can replicate the genome, which will make our positive sense RNA. And this positive sense RNA can be transcribed and it can be replicated using the RDRP, that RNA-dependent RNA polymerase. And again, for some of this, I'm not expecting you to memorize everything. I know it's very complicated. I want you to kind of understand the general way that this works. The way that I'll ask questions is going to be more focused on your understanding of the scenario and less on memorizing the exact details between each of them. We have retroviruses. These are viruses like HIV. They have a positive sense RNA strand, and it uses reverse transcriptase to actually make cDNA, which is going to be integrated into the host DNA, making a provirus or prophage if it's in a bacteria. And that can be transcribed, making new mRNAs, right? That can be packaged into new viruses, or just they can remain latent over time. In your double-stranded RNA genomes, you've got both forms of RNA. So one of them is going to make your mRNA, right? So the positive RNA strand can be directly translated, and it can also code for a RNA-dependent RNA polymerase to replicate the genome for packaging. All right. So that's a lot of different options. We're going to talk a little bit about how these genomes can change over time. They usually have a faster rate of genomic change than living organisms. Part of this is because they replicate rapidly and they produce a lot of virions, right? So there's probably something like 10 to the power of 31 virus particles on Earth, and so there's a lot. And so each infection can produce like many individual, or each cell that's infected can produce many individual virions. And these, and if the genome happens to be made of RNA, they typically mutate more than DNA. And that's because, as we talked about before, DNA polymerases have a proofreading capability, but RNA polymerases do not. All right. They can produce neutral, beneficial, or deleterious effects. And any genetic change that limits infectivity leads to what is called an attenuated strain. These are what we use in many virus vaccines, but not all. Beneficial mutations may allow the virus to escape the host immune detection, broaden its host range or host tropism, or expand its cell or tissue tropism, overall increasing its infectivity. You can get reassortment. Now, this happens when you get infection from two strains of the same virus. And then during the process of assembly, some of the genetic material from each of the virus strains is incorporated into the new virions, leading to a new viral strain. So that brings us to the ideas of antigenic drift and antigenic shift. Um, this is really important when we think about flu, which has the HA or hemagglutinin and NA neuraminidase spikes. And these spikes are antigens against which the immune system can create antibodies. So these are the things that the immune system attack. Uh, and so influenza has an RNA genome, and it causes it to mutate, fairly frequently. And that can lead to minor changes in the hemagglutinin and neuraminidase spikes. And so that slow change that through mutation is what we call antigenic drift. So you're not getting full scale change in your spike proteins. You're just getting these small accumulation of mutations over time. And that's the drifting. And so think of that as, to me, drifting is a slow process. So if you're drifting out in the water, you're very slowly moving. If you're antigenically drifting, your antigens, which are in this case are the spike proteins, are drifting very slowly. Now, occasionally you can get antigenic shift. So this is going to be when you get like infection of an organism through where you get two different strains of flu. In this case, we have a bird flu and a human flu that both infect a pig. And so this pig flu, or this pig that now has two types of flu, allows for the reassortment of the genome, and you get this new strain of the virus. It doesn't always have to be highly virulent, but it can be. And that is ways that in the past we've had highly virulent pandemic strains of flu. And so antigenic shift can lead to increased infectivity or expanded host range. There's no residual immune protection from infections or vaccinations because you've got this reassortment. And then this can potentially lead to a pandemic. So with that, you should be able to differentiate between types of viral genomes and explain the central dogma as it applies to viral genomes. I hope you enjoyed, and I will see you soon.